Since the dawn of time, humankind has produced nearly 4.5 billion metric tons of plastic waste. Now, much of this plastic has ended up in landfills, contributing to the surfeit of waste that we see today. And in response to this waste, institutions like the University of Wyoming have moved towards using biodegradable plastic alternatives, particularly those made out of PLA, or polylactic acid. Now, the trouble with PLA-based bioplastics is that many of them end up in landfills like their plastic counterparts, where they degrade under suboptimal conditions, producing mass amounts of methane and contributing to global warming. The alternative to putting PLA-based bioplastics into a landfill is composting them, which, through the use of microbial communities capable of degrading PLA, produces far less methane. Now, the University of Wyoming's Agricultural Community Resource for Everyday Sustainability, or Acres Student Farm, if you will, has tried taking PLA-based waste from the institution and placing it into stage 2 compost to measure degradation. They did, however, notice that limited degradation was observed. Our goal then is to better understand what soil treatments may be necessary to stage 2 acres compost in order to better degrade institutional waste made of PLA. In particular, we'll be examining pH, moisture content, and temperature for its effects on microbial communities' ability to degrade PLA naturally.
On phase 1 so far. And as you recall, phase 1 is the isolation and identification of PLA degrading microorganisms in acres compost. So we began by performing an enrichment of the bacterial communities in acres compost. And we got ample growth in the enrichment. So we plated those organisms onto M9.5% glucose PLA plates, with the hope of identifying organisms that can degrade PLA, but giving them another carbon source to help them get started. And we did observe growth on these plates. So we moved on to M9 PLA. So in this case, PLA is the only carbon source on these plates. So we took colonies from the 0.5% glucose plates and plated them onto M9 PLA and observed growth again. So we took those colonies and plated them onto Schadler blood agar to isolate Bacillus species and Pseudomonas agar to isolate Pseudomonas species, both of which are known to degrade PLA. And we have observed growth, ample growth on both of these, showing us that we have isolated both Pseudomonas species and Bacillus species present in acres compost capable of degrading PLA. In phase 2, we seek to identify the physiochemical conditions most amenable to supporting endogenous microbial populations in compost to degrade PLA. So we are examining 3 different conditions: water content, pH, and temperature. And to do this, we have set up different treatment groups using sterile sample cups filled with acres compost. And we have adjusted each of those to one of 3 water contents, either 40%, 50%, or 60%. And then each of those treatment groups was broken down into 3 pH treatment groups, either acidic using citric acid, unadjusted, or alkaline using lime. And 2 identical treatment groups were then broken into temperature groups, One was incubated at 30 degrees Celsius and the other at 37 degrees Celsius. And 10 PLA granules were added to each of these sample cups, and the precise weight was determined and recorded on the outside of each sample cup, so that at the end of the incubation we can take out the granules again and weigh them precisely to determine the weight difference due to degradation. So far we have set up the treatment groups and are aerating them every 3 days, and we hope to see what happens in a few weeks.